Hi, everyone. Good morning. All right. Happy Thursday. It's Thursday, right? It's happy Thursday. Hope everyone is doing well. It is, it's been really nice here the last few days in Michigan, and now it's raining. So expect rain soon, I guess, because it seems like every time, whatever weather we have ends up being the weather you guys have shortly thereafter. So, oh, thank you, Brittany. Brittany says I look nice. No, I just didn't pull my hair back. That's all. I pull it back all the time. Sometimes you just need a change, right? Like let it be free. Let it be free. Thank you. Wow. Oh, I should pull it back so you guys aren't distracted. All right, hold on. No, I'm just kidding. All right, what should we do? Oh, okay. So let's do a little housekeeping first, and then I'll pull up the syllabus. Where is it? Okay, well, here it is here, but I gotta show it to you guys. Oh, come on, Zoom, work with me. Here we go. All right. I'm gonna share the syllabus. All right, so you guys should be able to see the syllabus. Let me fix this thingy. These. Cool. Yeah, so wow, we are, whoa, how far we've, oh, let me change, oh, get out of here thing. Get out of here. How far we've come, how far we've come. The last time you guys had your lecture exam 2, most people did really well, surprisingly well sometimes. Little questioning that. May need to adjust some of the format for lecture 3 to, yeah, but overall, yes, everyone did very well for lecture exam 2. I have uploaded your grades for lab exam 1 onto Blackboard, so your grades on Blackboard now reflect that. I haven't updated attendance yet, so attendance doesn't show anything for April quite yet. But also remember, attendance really doesn't matter that much. I mean, yes, it's worth 5% of the course grade, but like overall, if you're doing well in the course, or you're doing overall, however you're doing in the course is ultimately not going to be determined by attendance. So don't get all bent out of shape over attendance. If you're doing really poorly in the course, don't think attendance is going to save you. If you're doing really well in the course, don't think attendance is going to hurt you, right? Because your overall, the attendance is very small for what the course is worth. Let's see, today, all right, so if we look at the syllabus right now, it says that today's class and then next week's classes are all about angiosperms, and that's good, and but also yeah, it's not a lot of material left to cover, so we'll see how we do today. There's a chance we, depending on how today goes and the next class goes, we may yeah, we just don't have that much material left to cover. And then for these two, the 27th and 29th down here, right now on the syllabus, it says plants and people, and then it says urban plants and plant-based careers. Whoa, hi, Rosamund, you have a question? Hello.
I'm talking to myself. I have a bill and I'm trying to pay it. I'm sorry.
Pay your bills, Rosamund. That's important.
Thank you, Professor.
Okay, all right. Don't forget to pay your bills, guys. That is important. Yeah. Anyway, so on right now on the 27th, it says plants and people, and then 29th says urban plants and plant-based careers, right? That's what that says here. So this semester, I'm doing something a little different for those classes in that they could even be renamed. But basically on those two classes, for each of those classes, we're gonna have special guests, and I've lined up some awesome special guests for you guys. 3 on the 27th, and then 3 on the 29th. These are people who are very successful in whatever they do. They represent different parts of what you can do with a career focusing on plants. We'll have some academics, obviously, right? We'll have a couple PhD, we'll have a PhD student, we'll have a couple professors who study different parts of the plant kingdom and different applications. We'll also have some pretty awesome community scientists. We'll have a botanical illustrator and a freelance scientist, if you can believe that's a thing. It is. So that's gonna be awesome. So I'm looking forward to those classes. It's taken a lot of my work to set that all up. So I'm very much looking forward to those classes. And then the semester is over for us in terms of lecture. And we'll have our final exam. I think the final exam should be posted by now. I haven't looked. Did anyone happen? You don't have to look. But did anyone happen to look at when our final exam is? If not, I can look later. But the point is, then we have the final exam, then the semester is over. In terms of lab, they are cool. Catherine says they all sound so cool. They are cool. And they're all most of them are dear friends of mine. Let's see, this lab tomorrow you're going to start angiosperms. Maybe you'll finish that the following Friday, and then 2 weeks from tomorrow you have your your laboratory exam 2, which covers all the seed plants. So that's what's on deck. Wrap your head around it. I know you have a lot else going on, like other classes, life stuff, kids, bills, right, Rosamund? Bills. So, but that's
yes,
oh, okay. Thank you, Brittany. All right. So we have a final exam date is May 11th. Ooh, 8:30. Ooh. May 11th, 8:30 to 10. Sorry, 8:30 to 11:30. So, I'll confirm that, but I believe that's correct. Thank you, Brittany. What is that day? What day is that? Let's look. May 11th is a Tuesday. Yes, a Tuesday. So what does that mean in terms of the course? So that means, hold on, guys. Go back up here. So we'll finish all the angiosperm content by 22nd, the latest, which means you'll have 1 week, 2 weeks, 2 weeks and 1, 2, 3, 4, 2 weeks and 5 days to study the angiosperms, and it's not that much information. So everyone should do really awesome on the final exam, which covers angiosperms and the core course concepts. But just for your, for your schedule to look at. So you'll have the lab exam on the 30th, and then that means you'll have 1 week and 2 weeks. So sorry, 1 week and 1, 2, 3, 4, 1 week and 4 days after the laboratory exam until the final exam. Don't forget, okay, so people are like, so much time. I'm gonna forget everything. Oh, I'm gonna miss this class. Oh, that's nice. Well, you could always email me, and we could always meet up one-on-one. Tell the department if you like this. Oh, that's the other thing. All right, so I'm getting distracted, obviously. So in terms of missing this class, this may be the last time I teach this class, guys. I don't know, because basically, yeah, I mean, I thought the last time I taught this class in spring 2020 was going to be the last time, because I moved to Michigan. Remember, I'm in Michigan, and so I'm assuming next spring is going to be in-person classes, I would assume, right? And so I can't fly to New York, New Jersey to like do this class. So if you like this class, I'm going to be sending out the SIRS, that's this the student instructional survey thing, the SIRS, I'll provide a link or a QR code for that at some point, maybe next class, for you to evaluate this course. And thank you, Miriam. Miriam's like, I'm going to be recommending this class to everyone. Well, hopefully you recommend them to a class that exists. Hopefully this class exists. So when I send you out these surveys, make sure you are honest in those surveys. I really look at those surveys very closely, and I've curated the class. This is the fourth time I've lectured this course, and I've curated the class based on student feedback. So please, when you do get that survey link, be very honest, but also if you really do like this class and you don't want to see Plant Kingdom die or go away, because it will, please be vocal about that in the survey as well. Because it's, yeah, I don't know, unless I can figure out a way to like keep teaching this virtually next spring, but I don't know how people are gonna feel about that. So we'll see. Another thing that came up in the chat box was about the exam. Ooh, so early, ooh, right? Yeah, we will not be meeting at 8:30, I'll tell you right now. In the past, when I do the final exams, even though they give you that crazy block of time, like 8:30 to 11:30, first of all, whoever's having exams that long, just stop, just stop. No, but for final exam, the final exam will run like the other exams we've had so far, meaning that we will not meet in person, or we will not meet on Zoom. Even though some of you keep on trying to go onto the Zoom during exams, I don't get that, but maybe you just want to see me. I get it. Maybe I get that. No, I don't get it. But we won't actually go to Zoom on the final exam day. You guys will just take your final exam, and the final exam will happen at the exact same time that our normal exams happen, not 8:30, 10 to 11:20, right? So you're welcome. All right, let's get started. All right, all these questions. People are there's a question, is there going to be a curve? No, because many people in the class are scoring. So a curve, the purpose of a curve is to account for professor error, as it were. So like let's say I give an exam and the highest grade is a 90, right? I did something wrong then. If I gave an exam and the highest grade is a 90, out of 40 students, obviously there's a disconnect there and I did something wrong. Hence, you would do a curve and make that 90 100 and everyone else would be bumped up a letter grade, right? Well, in this class, the majority of students get A's, and a lot of students sometimes ace assignments and exams. And therefore, there's no justification for a curve, because what do I curve to is the way to answer that. Hira is asking, can you open the results for test 2 so we can see what we got wrong? Yes, but not right now. But yes, I will. Open exam 2. I will. Just keep in mind. Oh, lab exam 1. I was gonna say lecture exam should be open. All right, and also lab exam 1. Yes, I'll do that later. Keep in mind though, right? Keep in mind that the next lab exam and the next lecture exam do not include that material. So I'm happy that you want to see what you got wrong. And obviously if either Bruno or I made a mistake in our grading, please draw that to our attention. But they're not cumulative, right? So keep that in mind when you're looking those over. Like the next exams are not cumulative. All right, we got to get going. So let's do this. I'm gonna stop sharing the syllabus. Oh, there's one more thing I want to say about the syllabus. Go back. Share. All right. Yeah, here, you guys can see this. I'll go up. Don't cheat. Stop cheating. Don't cheat. Don't plagiarize, right? Don't cheat. Don't plagiarize. I understand we're on Zoom right now. And I understand when you're taking the exams, you are are not being watched by me, right, the entire time, or at all, really. So I get that. And like, I've been nice in that I know that virtual classes aren't easy. And some are harder than others. I don't know where this one falls on that spectrum, but the point is that I know that you guys didn't intend to have virtual classes. And so I don't want to make your life harder than it already is. So when you take exams, for example, I know you're going to look at the slides. I know that you might even try to pull up a recorded lecture, which please don't do that because you might crash your system. And also each one is like an hour long. So I don't know how you're going to benefit. But anyways, I know you guys do that. I know you're looking at your notes and I'm okay with that. But don't be like assisting each other. Like it's pretty obvious when some people are assisting each other. It's also this last exam. Some people who haven't been doing great at all have all of a sudden done super hot on this last exam. So I'm gonna keep that in mind and just know, oh, I'm starting to get my professor face on. Like just know that there are tools on Blackboard that I could use to ensure that you guys don't cheat. For example, I can make you do an exam on Blackboard where it freezes everything else on your computer. You won't be able to open a browser. You won't be able to open a document. That's an option on Blackboard. Another option on Blackboard is that you have to take an exam with your webcam on. I have not opted for these things because I want to like create a positive learning environment, right? I really want you guys to have fun in this class. I want you to enjoy the material. That being said, do not cheat. Don't rely on your friend. If you're the friend that's helping your friend cheat, you're doing yourself a disservice because if you get caught, you're going to be in trouble. And if you're the friend who's asking your friend to help you cheat, that's not nice. Don't do that. Like, be responsible for your own life. Be responsible for your academic pursuits. Don't cheat, all right? You owe yourself better. And like, if you get an A in a class from cheating, and then you're like, family's like, oh my God, we're so proud of you. And it's like, you're going to know that you half-assed it. So yeah, that's my little speech. I will be adding some questions to the final exam that are short answer and or essay. So yeah, don't cheat. Don't cheat. Okay, sorry. I had to get that out. It was bothering me. All right, we're gonna move on. We're gonna move on to Angiosperms Part 2. Let's go here. And we'll share the screen. All right. All right, cool. Hopefully you guys can see the screen. If you can't, let me know. Let me pull this big. All right, so we might not get through all of this today, which is fine because I only have this lecture and then this presentation and one other presentation for us to get through, and we're ahead on time. So we'll see how we do today. I don't want to rush. Take our time. Okay. So Angiosperms Part 2. Before last class, you had your exam. And before that, you had a day off to study. You're welcome. And then before that, we had an intro to angiosperms and flowers, right? And so today we're going to talk more about angiosperms. In particular, we're going to talk about growth and then evolutionary lines in the angiosperms. All right, so just an FYI, and I think I mentioned this in a previous class, the angiosperm phylum name that we're going with is Magnoliophyta, right? And we even talked about the plant Magnolia, which is where the name Magnoliophyta comes from. You'll see another name, especially like I said, the lab material. You'll see another name, which is Anthophyta. But that term is now outdated. And I haven't changed it in the lab because it's a good opportunity to show you the dynamic nature of science, in that we, as scientists, we do our best on a day-to-day basis. But sometimes we learn more information. And when we learn more information than we previously had, we may need to adjust. And so hence why the name used to be Anthophyta, but then we learned some more stuff about plants and we changed it to Magnoliophyta. We'll talk more about that in this lecture, or maybe not this lecture, but yeah, this lecture, I think. All right, so primary growth, This slide should look familiar. Oh yeah, it's a recall slide, right? So we've seen this before. But just as a reminder, right, primary growth comes from the root and shoot apical meristems, and those apical meristems give rise to those primary meristems. We've seen this before. This is a close-up of the shoot apical meristem. Oh my gosh, move thing, move out of the way. There we go. This is the shoot apical meristem right up here, right? This is a close-up of it. Here is your apical meristem right there. And notice all of these primary meristems being created via the division of the apical meristem, right? So like here is your procambium, that's a primary meristem. Notice I'm using the same color as the word primary meristem. So ground meristem, sorry. Another primary meristem, and then the protoderm, right? Those are the three primary meristems that arise from the division of the apical meristem. We know this already. We know this already. Oh, look at that. Thanks, me. And we already learned about secondary growth. The lateral meristems, pretty much everybody got this right on the, so I noticed in the last two assignments, I obviously really want you to like get this right? Because like in the last two assignments, there was a question about lateral meristems and most people got them right on both assignments. So that's good. But yeah, there's the vascular cambium and the cork cambium. I did notice on the exam that some people are confusing where these things are, like in the stem, which is surprising because we've talked about it several times, and also you would have had it in lab as well. So if you're confused about where these are in the stem, go back to conifers, go back to ginkgo even, go back to your lab material and figure it out. For example, some people think the vascular cambium is like really close to the edge of the stem. No, it's not. So yeah, if you have questions about that, please let me know if something is unclear. But yeah, so you have secondary growth by lateral meristems. The vascular cambium gives rise to the secondary xylem, secondary phloem, and the cork cambium, which is the lateral meristem toward the outside of the stem, gives rise to cork and Phenoloderm. Oh, and then we have this nice, I say ugh, but it is a nice summary slide, because it really shows you how you get from, one cell type to the other. Oh, so Vivian says, I confused them, LOL. So Vivian, what, is there something I can do to, so it's really about, what I would suggest, if you're confused about these meristems, what I would really try to do is compartmentalize the meristem name with what it makes. And if you do that, it may help you stay oriented of like where the meristems are. And this works for both the primary and secondary meristems. For example, if you're just talking about primary meristems, if you think to yourself, okay, protoderm, what is protoderm? And you can recall that it makes epidermis, epidermis is toward the outside of a young stem, right? It's the skin. So obviously, like the protoderm is not going to be buried on the inside of the stem because it gives rise to the skin. So it has to be relatively close to the skin. But a better thing in terms of the lateral meristems, where did those, here, which is, here, let me use, I use yellow for those. So, which is your cork cambium and your vascular cambium. Again, think about what these things actually make. Cork and Phenoloderm. So remember last time we talked about like bark and stuff like that, like cork and Phenoloderm and cork cambium and secondary phloem, but like those are all parts of bark, like bark on the outside of a tree, right? So cork cambium is giving rise to those external structures that you can see with the eye, and then the tissue right below that. So that's going to mean the cork cambium is closer to the outside of the stem. Whereas vascular cambium is giving rise to secondary xylem and secondary phloem. And remember, like when you look at an old stem, like think of a big trunk of a tree. If you were to cut that down, almost all of that tissue is secondary xylem. So it's toward the middle and all the way toward the outside, but not the outside. So just try to maybe that'll help. But either way, yeah. There's also some nice diagrams from the other. Vivian said, "Oh yeah, that makes sense." Yeah, there's also other diagrams to refer to as well. Let me get out of annotate. Okay, let's move on. We're really sluggish today. That's okay. I think I'm stalling a bit because we don't have that much more to go through. All right. This is a recall. Not going to go through all the things. Just note that all plants make primary growth. Only some plants make secondary growth. And for those plants that make secondary growth, you would have originally had primary growth. And so the main point of this slide is showing you, here, I'll use the same colors. Remember, primary growth is for lengthening at the tips, at the root tips, and the shoot tips. Notice, look, here is an apical meristem, right? Look, here's an apical meristem. This one will grow into a branch for lengthening. This one will continue the stem on for lengthening. And then look down here. Each of the root tips also has apical meristems to lengthen the roots. Lengthen. We already know this, but just know. So look, this plant on the left has primary growth via the apical meristems at the tips, but this plant is starting to widen and get thicker, which is what secondary growth is for. And so inside the stem, we see the vascular and cork cambium to allow the stem and later, or to allow the stem, but also the roots. See those red and blue lines going through the stem and roots? That's this. Red is the vascular cambium, blue is the cork cambium. That's what's showing you on that inside of that stem. And if you were to do a cross section through that, here you go. Look, vascular cambium. What color? Yeah, vascular cambium is this red line in here, giving rise to this secondary phloem right here, and secondary xylem, which is the blue thing here, right? So the vascular cambium is the red circle in this cross section. Cork cambium is the blue, dark blue circle. Whoops. Yeah, so there you go. So I was just saying. Yeah, so let's move on. All right, so we already know this. This is all recall, right? The procambium, which is a primary meristem, produces the primary xylem. And then because today I think we're going to talk about xylem. So the procambium produces the primary xylem. And then the vascular cambium produces the secondary xylem. Yes, that's right. We're going to talk about xylem cells today. So xylem is a cell. Xylem is a cell, and if you have a lot of xylem cells, you have a tissue of xylem, right? Part of the vascular tissue. And the main cells in xylem are what are known as tracheary elements. Tracheary elements. And if we were to look at each of those tracheary elements, we would see two types of cells. There are tracheid cells, and then there are vessel element cells. For both of these cells, so for both tracheid cells and for vessel element cells, both are elongated, both are dead at maturity, and both may have pits, which we'll look at in a slide coming up, to help with flow, So dead at maturity. That might be a new idea for you to wrap your head around, but like basically, okay, I got it. This thing is just popping. Quiet thing. Yeah, so dead at maturity, we'll talk more about that, but basically it's what that means is that when the tracheids are mature and the vessel elements are mature, they will also be dead. So this is kind of what I was talking about before. Like if you have a wooden, we're talking about xylem, right? If you have like a wooden table or like a wooden floor or wooden door for that matter, that is made up of wood, but obviously that's not living wood, right? It's dead. It's dead wood. That's why you can have a floor of it. And that's because those wooden floor and a wooden table are composed of xylem, and the xylem cells are dead on maturity. And so when we cut it down and make a table out of it, it's still dead, right? It's still dead. Right. Some plants only have tracheids. So the plants that only have tracheids are your spore-releasing vascular plants. So your ferns, lycopods, horsetails, and the gymnosperms. Those plants only have tracheids, no vessel elements, only tracheids. We saw this slide during the conifer lecture, and just so you know, right, this is showing you, so depending on how you cut through a conifer stem, you will see different views of the tracheids, So each of these cells, I guess I'll use, let's see, what color should I use? I'll use blue. So right here, let's go to the side, or actually we'll stay at the top because we're already at the top. So each of these squares, this is going to be fun. Even these, all right, well, so here, first we should do this. First, let's get oriented. We are in secondary growth here. So this is our vascular cambium right here. It says cambium on the slide, but that's vascular cambium. Okay. Vascular cambium is going to produce secondary xylem toward the inside. And so each of these squares, there's one, is the top of a tracheid, right? Here's a whole another row here, right? You see it says early wood. Remember early wood and late wood. This particular piece of wood doesn't have late wood yet. Well, actually, this is all late wood, but don't worry about that because that's from the previous year. The point is that each of these things is a tracheid, and depending on how you cut through the wood, you'll see it in different ways. So if you do a cross section or a transverse section, you'll just see the tops of the cells. So you can picture like a straw, like a drinking straw. So if you were to cut cross section through a stem, basically seeing the top of the straw, right? Like a circle, but in this case, it's a square. If you were to do a tangential section, you would see the tracheids long ways, right? So like this is one tracheid. Here's another one right next to it. And lastly, if you were to do radial section, come on, slide, advance. You would see a combo, because you would see the tracheids that are like this, like the tall vertical tracheids. Right, here's another one. Sorry, he's having a hard time. All of these are them, right? All of these are them. But you would also see some tracheids that run horizontally like this. This is one, whoop, which make up those wood rays. Why am I telling you all this? Well, it's just good to know that depending on how you cut something, it's going to look different orientations, right? So like for those of you who are going into medical fields, or you're going to have to be like performing surgeries and things like that. Obviously, if you know, orientation matters, things are going to look differently depending on how you cut through things. That's why I'm showing you this. Another reason this is useful is a lot of times people talk about wood grain, right? Oh, I like the grain of this wood. etc. That has to do with the way that the wood is cut and what tracheids are visible, and also the species of the wood, but still, it has to do with this stuff. All right, and this is what I was just saying on the previous slide. There's two orientations of tracheids in secondary xylem. There's a vertical orientation, which are the ones going up and down, like I already said. This is the here's a vertical tracheid, right? Here's the top of a vertical tracheid, right? And then there's horizontal tracheids that make up the rays, right? And that's what that says here. These slides will help you in lab. So I'm giving you some pointers there. These are real microscope slides of what the drawing on the previous slide showed us. So for example, here it says tracheids of a transverse or cross-sectional view. So that's here, that's this view, but in a real specimen. So remember, I told you if you do a cross-section or a transverse, you're only going to see the tops of the tracheid straws, as it were. And so yeah, look, each of these circles, and each of these circles is the top of one tracheid, right? All of these circles, right? This is all secondary xylem except for the resin duct. So that's what it looks like, a transverse or cross section. If we were to do a radial view, right? So that's this view, radial section for wood, you would see both vertical and horizontal tracheid, right? So here is the vertical ones. Oh, there's a vertical one, right? It continues down all the way here. And here's the horizontal ones going this way. And think about it, guys. Like, okay, so tell me in the chat box or shout it out. What does xylem even do? What does xylem even do? What is the point of xylem? Just to like be dead? What does xylem do, guys? Transfer water, transport water. Yeah, it moves water, right? It moves water. And so you need to, where's the plant getting water? Where does the water come from? If you're a plant, this is not a trick question. What part of the plant absorbs water? Oh, thank goodness. Yay, everybody's saying root. Yes, right? Water is absorbed at the roots, but then the water needs to go to the rest of the plant. It needs to go to the stems. It needs to go to the leaves. It needs to go to the flowers. It needs to go to all the parts of the plant. Hence, tracheids. So tracheids are like big straws that are pulling the water up from the roots to the rest of the stem, to the rest of the plant. But if you get super big and wide via secondary growth, you also need to move water horizontally. And I'm pointing that out on this slide because that's with this radial view, you can see these vertical tracheids are going to let water move up through the plant from the roots. And then these horizontal tracheids are going to allow water to move horizontally through the stem. That's why I brought that up. Does that last view, that tangential view, so this view right here, for me, the easiest way to tell if you're looking at a tangential view is to look for the ends of the radial of the horizontal tracheids. So for example, like I told you, this is a wood ray, right? With horizontal tracheids. Well, guess what? This is a wood ray too, but we can only see the top of it, right? We can only see this part of it right here. Look, here's another wood ray, like coming at you, right? Coming at you in this tangential section. So that's what I always look for when I'm looking at it, looking to tell like what orientation am I looking at it, right? So like here, look, you can see the rays coming at you. So this is just to keep it all together, this is what I was just drawing on that previous slide, right? Look, there's one, there's one, etc. And so those are the horizontal tracheids coming at you, right? Like coming right at you. And then these are again all the vertical tracheids. Okay. Okay, I mentioned that tracheids sometimes have pits. So what are pits? Pits are holes, basically. And these pits allow the water to pass from cell to cell. So when their tracheid has a pit, there is a thin membrane that covers the hole that allows the water to pass through. And the water only moves sideways from cell to cell, not upwards. I'll show you what that means. Here is like an example of pits. So you have this is a cross-section view or transverse view on the left, and then a radial view on the right. And this is a close-up of the tracheids. And these circle things, I'll use a different color, orange. These circle things are pits. Here's a pit. Here's a pit. If you're looking at a cross-section or transverse view, you can see the pits here. And so here on the radial view, this is a vertical tracheid. You can see all the pits on this vertical tracheid. And it says water only moves sideways from cell to cell, not upwards. What that means is that the way water is going to move from this tracheid to the adjacent tracheid is through this way. So like the water will move from this pit to that pit, right? Or this pit to that pit, or this pit to that pit. The water won't enter a cell above it, right? It'll be transferring water to a cell next to it via these pits, right? Sideways. Likewise, if we're looking at this cross-section view, this tracheid is going to pass water into that tracheid via that pit. This tracheid will pass it via that pit, etc. Right? Just to hit this home, just to hit this home with the best one. Yeah, we can do here, right? So like if we were looking at this radial view, here's a pit, and this tracheid is going to pass water to that tracheid sideways, right? Oh, that's not good. Right, here's another one. Look, this tracheid is going to pass water sideways to the other tracheids. But as it moves sideways through these vertical tracheids, it will be moving upwards, but I'm just saying it passes from cell to cell sideways through the pits. Where's the pit in the cross section? It's harder to see because you're only looking at the tops of the tracheids. But if you were to zoom in, like you could look, so you could see down here, like there's a pit on that vertical one, but because when you're looking at a cross section, you're usually only seeing the tops of the cells. More often, the pits just look like these little bulges in between tracheids. And in fact, Brittany, I'll show you here. That's what this is showing you here. So this on the left is a cross-section view. Cross-section, AKA transverse view. And I'll use the colors I used before. Like here is the top. They're not always square. They can be, like I said on that drawing, all the tops of the tracheids are squares, but they could be, they don't have to be a square, but like each of these is the top of a tracheid, right? Or think of like a straw. This is like the top of the straw. And if you notice between each one of those, or some of them because of the slide, you see these little bulges, and each of these bulges are pits. So that's what it looks like on the cross section, AKA transverse section. It's a lot easier to see the pits on a radial or tangential section. No problem. Cool. All right, so let's move here. So in the angiosperms, which are, the ultimate of plants, right? They have flowers. Tracheids have evolved into vessel element cells and fiber cells. So don't get dizzy, but I'm just showing you. Remember I said some plants only have tracheids. spore-releasing plants and the gymnosperms have tracheids. Spore-releasing plants and gymnosperms have tracheids. Boom, boom, boom. Angiosperms no longer have simple tracheids. They now have what are called vessel element cells and fiber cells. And these are specialized water conducting cells that move water more efficiently than simple tracheids. So, but they have some similarities to the tracheids. So for example, the vessel element cells, which will be color coded going forward in green, are also dead on maturity. So they're non-living, just like the tracheids. But a difference is that now at the top of the vessel element cells, they have what are called perforation plates, a plate. Oh, sorry. So it's top and the bottom. They have a plate. And that allows water to flow upwards, not sideways. So that's great because it's kind of like a slower process if you were trying to move water from the roots to the stems or from the roots to the top of the tree. And each cell has to pass the water sideways to the next cell, sideways, 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 sideways. It's kind of like going up on a 100-story building, taking the stairs, right? Because you're kind of like, okay. I mean, like, yeah, you'll get to the top, but it's not super efficient. What you can think of is like the vessel element cells are kind of like the water taking an elevator, right? Because you don't have to go boom, 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 boom anymore. You just go whoop, straight up, right? Upwards. They do still have pits that allow water to move sideways. So they haven't lost that ability, but they can now move water upwards. So here is what a perforation plate looks like. There's different types of perforation plates, depending on what species you're talking about. Here's an example of a simple perforation plate between two vessel element cells. Remember, these are all cells. Oh, I should go back. What color did I? Yeah, green. Remember, all of these cells that we're talking about, tracheids, vessel elements, etc., those are all parts of secondary xylem, just so you can keep it organized in your brain. So this circle here is a perforation plate, which is at the very top of a vessel element cell. And you can see here down below, you still have pits, right? Look, there's still pits to pass water sideways. But which do you think is more efficient at moving water? These little pits or like this big boy, right? Like move that, get that water up there. We need the water. Go, go, stat. So perforation plates are better at moving water. Right, note the cell still contains pits. Here's a more complex perforation plate. This one has almost like a ladder-like opening, right? So this whole thing is a plate. This whole thing is a perforation plate in this vessel element. You can still see, here's some pits on the side, right? More complex. Right, note this cell still contains pits. Yes. Okay, so there's pros and cons. There's pros and cons to having tracheids and having vessel elements. Like neither one is perfect. There's pros and cons. So the vessel element cells, which have those perforation plates, the pro is that they're more efficient, right? You don't have to pass the water side to side to side if you don't want to anymore. Now you can just go straight up. So that's more efficient. So that's a pro of the vessel element cells. But it's way more risky. It's way more risky than passing water sideways through pits. Because number one, vessel element cells, you might have noticed these perforation plates, there's no membrane. This is just a giant hole. This is like a hole with some, this is like this is like a grate in the street, right? It's like a giant hole with some little like lines going through it. This one doesn't even have the lines. This one is literally just a gaping hole. And yeah, that's really efficient at moving water, but there's no membrane anymore to kind of regulate what's going through from one cell to another, as there were with the pits. The pits have a membrane. Vessel element cells don't. And so there's no regulation about what can move through. So that could promote things like, that could allow pathogens to move very quickly from cell to cell to cell, because they don't have to pass through a membrane anymore. So that's a little bit more risky. And then on top of that, you can have air bubbles form, which would prevent water from moving upward through that cell anymore. If you had a tracheid cell, the air bubble would stay within a single tracheid cell. So I'll draw it. I think I need to draw it. Yeah. All right, so let's do tracheids. I use blue. Here's trachea. And here's another trachea next to it. Sorry for my drawing. Here's vessel element. And here's a another vessel element. And just for purposes of this, well, I'll draw another 2 more tracheids on top. Right quick, like one here. And I'll draw more vessel elements. Here's one, right, here's one. Okay, here's one. All right, so air bubbles. Let's use, I'm gonna use red to indicate an air bubble. So let's say an air bubble forms in this vessel element. There's an air bubble. Now, water, all right, so which way is water moving through the holes of these perforation plates through the vessel elements? Just straight up, right? Upwards. But if there's a giant air bubble there, up, no good, over, can't move anymore, done. Air bubble has stopped me. I can't move my water to the next one anymore. Whereas with the tracheids, let's say air bubble forms right here. Since tracheids move water sideways through cells, through pits, they can avoid dealing with this air bubble. So this trachea is going to be moving water from this to this, and from this to this, sideways. So if an air bubble forms, who cares? Because it's just the water's still gonna be moving sideways. The air bubble will be at the top of the trachea, and the water can just still keep moving through sideways from cell to cell. But it's slower, has to go through a membrane and move side to side. So anyways, vessel element cells, the pro, more efficient, the con, more risky. More risky. And I think we'll talk about this, and then I think we'll finish for today about this. The next thing that angiosperms have is these fiber cells. So you have vessel element cells, which we just talked about in detail, and then fiber cells. Fiber cells, like the tracheids, are also dead on maturity, right? Non-living. And they have an extremely thick cellulose wall, which allows mechanical support. And this is why we call angiosperms hardwoods and conifers softwoods. And this, I think, was a question on an assignment or the exam, one or the other. Only angiosperms have vessel element cells and fiber cells. The other plants we've looked at don't have that. The fiber cells are extremely thick, and they make, because of this thick cellulose wall, they make the wood harder, and that's why we call angiosperms hardwoods and conifers softwood. So this is just a comparison of the different type of things that we've just covered about these xylem cells. So I'll use, right, so these are labeled for you. Right, look, A and B are examples of wide vessel elements. So that's this and this. Look at these, this one has this giant perforation plates. This one has a somewhat smaller perforation plate, but still big holes at the bottom, that bottom and top, that allow water to move upwards from the roots. C is also a vessel element, but it's just a little more narrow. And it has the perforation plates kind of get cut off in this picture, but they're most likely those kind of ladder-like ones. Next is D is a tracheid. Here's a tracheid, no perforation plates on that, just pits. And then F is fiber cells that provide mechanical support. And note these, all those spots on the sides are pits. Not sure. Yeah, those are probably pits too, but the fiber cells don't really serve so much for transporting water as much as they provide support. That's their main role. That's their main role. All right. This is the same. I don't see perforation plates on C. I know, I think it got kind of cut off in this slide, but I can kind of see one here. Don't worry, you'll be able to see it better on this drawing that's coming up. Here you go. So here we got vessel elements are on the right, and then the tracheid is on the left. Here's our vessel elements. You see the big, this is a wide one. Here's a slightly less wide one. And here is one that has perforation plates that are like a ladder. Tracheid is on the left. Oh, not purple. Blue. Tracheid is on the left. Notice no plates at the top and bottom, right? No plates, just pits, just pits. And all these are smaller pits as well. But yeah, so you can see here, there's still pits on the vessel elements, which in the case that there was an air bubble, perhaps water could still move sideways through these pits to another one. But ultimately, if you have a perforation plate, that's not the way you really want to move water, right? You want to move water this way. Okay. Yes. All right. So sieve tube elements, and then let's see. I think maybe. Remember, there's just this slideshow. Okay, good. Yeah, actually, we'll cover the sieve tube element. Sorry, we'll cover the sieve elements, and then we'll cut for the day, and that'll put us in good shape for next next classes. So we just talked about xylem tissue in vascular plants. Now let's talk about phloem tissue in vascular plants. So in the angiosperms, so going back, we talked about xylem. In the spore-producing vascular plants and the gymnosperms, we had tracheids. In the angiosperms, we had vessel elements and fiber cells. Now we're talking about phloem, and there are two types of cells for phloem cells. Which are collectively the sieve elements. Sieve. There are two kinds. There's sieve cells and sieve tube elements. So similarly to our tracheid versus vessel, similar to this, right? Vessel elements versus tracheids. Spore-producing plants and the gymnosperms have sieve cells. Angiosperms have sieve tube elements. Here are sieve cells, which we would find in like gymnosperms or the spore-bearing vascular plants like ferns. So sieve cells are living. That's different than than the xylem cells we just looked at, right? Which were all dead on maturity. Sieve cells are living, and sieve cells have little holes or pores that are really narrow, and the pores in this slide are blue. And because we're talking about phloem cells, they're going to be moving sugar, not water. And in sieve cells, the sugar is going to flow sideways from cell to cell, not downward. So let me back up a little bit. So I asked you earlier, what do xylem cells move? And you told me water. And I asked you in what, like, where does the water come from? And you told me the roots, right? Phloem cells are moving sugar. Where does the sugar come from? Where did the sugar come from? This is not a trick question. Someone like came by and poured sugar on the plants. Like, where do plants get sugar? Leaves, product of photosynthesis of the leaves. Yes, the leaves are using sun and water and CO2 to make sugar. So sugar is made at the leaves, and it's going to need to move downward through the plant. It's going to need to move down to the stem. It's going to need to move down to the roots. So sieve cells are going to allow the sugar to move sideways from cell to cell, not downwards. So it's similar to your tracheid cells in that, see, these, all the blue things are holes, and the sugar is going to move sideways from sieve cell to sieve cell, much like tracheids moved water sideways from cell to cell. But then you have sieve tube elements, which are what the angiosperms have. And they're similar to the vessel elements that we looked at. But some differences are they're living, not dead on maturity. They do still have these little small pores on their sides. But then they also have larger pores, which are located at sieve plates. Sieve plates. So we had perforation plates in the vessel elements. Now we have sieve plates in the sieve tube elements. Here's a sieve plate. And these are the smaller pores. And then if you're in a sieve tube element cell, which is what the angiosperms have, now water can move down, or sorry, not water. Now sugar, so I'll use the pink for sugar, can flow through those sieve plates directly downwards from the leaves to the roots and through the stems. And they don't have to pass it sideways anymore. They can if they want, but more efficient to just go straight down. Here is a drawing of the sieve cell versus sieve tube elements. Sieve plate, more compound sieve plate on the sieve tube elements. Here's some at the bottom as well. And then the sieve cell only has pores, so it can only move sugars sideways, right? So sugars is moving sideways through the sieve cell. Sugars, oh, nope. Sugars are moving directly downward through the plates, sieve plates of the sieve tube elements. And sure, they can do it sideways too if they want, but again, like I said, more efficient to just go straight down. Okay, so here's an interesting thing. This is a good place to stop. So hold on. So the xylem cells I told you were dead on maturity, which makes sense because if your job is to basically be a straw, you don't want to be full of all this like jello-like cytoplasm, right? Like if you're going to be a cell that moves water like a straw, you need to be dead because you need to be empty. That's the best way to move water. But sieve cells and sieve tube elements are not dead. They're not dead, but they do move liquid, which is liquid sugar. And so the way that they do this is they have gotten rid of some cellular parts. So like if you look back here, these sieve cells and these sieve tube elements are cells that do not have nuclei. There's no nuclei inside of that. So it's like, how are they alive? If they don't have nuclei, how are they being able to be alive? And so they basically have these special cells that are called companion cells and angiosperms that basically keep the sieve tube element alive. So here you go. This is good here. All right, so you got, this is your sieve tube elements that we've already looked at, right? I'll use the same color as before, right? Sieve tube elements, here's one of them. Whoa. Sieve tube element has a sieve plate right at the top. There's also pores along the sides, but no nuclei. No nuclei, but it's alive. How is it alive? Because there's these adjacent cells which are stained very, it's hard. They're hard to see, but trust me, I'm running out of colors, guys. I'll use purple, I guess. Right here and right here are these things called companion cells. Here's another one here. Here's another one here. Right, so this one is actually 1, 2, 3. 1, 2, 3 companion cells. Companion cells basically keep the sieve tube elements on life support, I guess is a kind of a way to think of it, because companion cells have nuclei and basically keep the sieve tube elements alive. The reason why this has evolved this way is because if you're moving sugar from the leaves through the rest of the plant, you can't really be encumbered. You don't want the sugar to be blocked by a bunch of cellular material. So sieve tube elements have evolved, gotten rid of all of that, but therefore need help to stay alive, and that's what the companion cells do. Cool. So you can see here the companion cells regulate metabolism in the sieve tube elements, aka keeps them alive. And this allows unrestricted sugar flow in the sieve tube elements, which are basically empty because they don't have any nuclei. So that's a better way to say it than all my words that I was trying to say. Okay. And there's a summary table of all these cell types at the end of this lecture. So we're not going to finish this lecture today. We're going to stop for the day. We will pick up on this and talk about different angiosperm plant types next class, which will be on Tuesday. So Tuesday and Thursday of next week, we'll finish the angiosperms completely. And then the following classes, we'll have our special guests, which I'm very much looking forward to. So if anyone has any questions, I'm happy to answer them here, or if not, you can send me an email and I'll follow up there. And I will unlock the exam and both exams later. Oh yeah, phenology assignment. Okay, yeah, let me pull it. So Beatrice is asking if you can send me phenology assignments now. Let's look real quick. Let's see, when was the observation period? I'm looking at the syllabus right now, hold on. Observation period ended. When did it end? I can't remember. I have to pull up the instructions of the phenology assignment. Yeah, I think it was this week too. Let me just pull it up real quick. Plant Kingdom. Let me look at this. Let me look at the assignment instructions. Phenology. Oh yeah, today. Wow. Oh man, can you believe it? Here. So this is the instructions for the phenology assignment that are posted on Blackboard. Yeah, so look, your observation period ends today. But you know, you could obviously still take pictures this week if you need to catch up. Yeah, so if you are done organizing all of your stuff for your phenology assignment, whoops, right? Like if you're done organizing your table, which is shown at the bottom here, and organizing your PowerPoint presentation where each individual tree is on one slide showing the progression of that tree. It is technically due on the 29th, which is the last day of class. But if you have it ready now, you can turn it in at any time. So yeah, send it along. Yes, it was six weeks of observations is what it was. Yeah, because there's some questions about in the chat box. Vivian said my trees still don't have leaves. Oh, LOL. I'm sorry. Hopefully it may be dead. Yeah, it's gonna depend a lot on things. The species of tree, where it's located, all that stuff. Here in Michigan, everything is budded out. I mean, we have leaves, we have flowers, we have it all going on. Yeah. Oh no, Catherine says my mom cut down a tree I was observing midway. Oh. Were you like, Mom? So it's funny, that's like a new thing. Instead of my dog ate my homework, it's my mom cut down my phenology assignment tree. All right, guys. Well, if anyone has any questions, I'm happy to answer them here. Yeah. Where is your tree? My mom cut it down. And if not, email me and I will talk to you guys there. Oh yeah, Ben, I'll see you at 1 for our meeting, right? Cool. All right, guys. Thank you, everyone. Have a good lab and have a good weekend.
Doug, this is what I saw.
Oh no, Roseman. Oh, bills, right?
Have a lovely day. Thank you.
Thank you. You too. Take care, everyone.